fundamental tension between the two-dimensional nature of the image information that we get from the retina and the solid three-dimensional geometry of the things in the world that we need to understand and interact with. And in this talk, I hope I'll convince you that what the visual system does with this tension is quickly resolve it in favor of representing real-world solid geometry and getting away from the two-dimensional patterns that we tend to think of. I've always tended to think of in advance.
Is that the one on the National Mall?
So Mike was saying
that's the one I know, the one on the National Mall.
Yeah, that's what I've seen too.
Yeah.
This could be a different one.
Yeah, this one I think was marked on the photo set as being in Georgia, maybe. So.
I have a video of the one on the National Mall, and it's quite striking as you pan around.
Oh, really?
Yeah.
Yeah. Well, I could have used that video. So Anitha already introduced the ventral pathway, and I'll just re-emphasize that sort of officially the ventral pathway is seen as going from primary visual cortex, or V1, through V2, through V4, and then into posterior parts of ventral, occipitotemporal cortex, and then into successive stages into inferotemporal cortex, or TE as it's labeled here. And actually, this diagram shows that there are three channels that go through TE, so the whole thing is a little more complicated than Van Essen used to draw it. And Anitha also highlighted the idea that when you're back in primary visual cortex, the receptive fields, the parts of visual space that cells are actually responsive to, are quite small. They're just fractions of the degree of visual angle. By the time you get to V4, receptive fields in the vicinity of foveal vision cover several degrees of visual angle. By the time you get to the end of the pathway down here, you've got very large receptive fields that some people claim can cover the entire visual field. They're bilateral, and they tend to be very foveal. Then the other quick bit of background is something that many people will be familiar with. This is an idea that goes all the way back to the 50s and T.O. Binford, but it's had many expositions by many famous people, including David Marr and Irving Biederman. It's the idea that objects would be represented in terms of their parts and the spatial arrangement of those parts. So the latest name for this that I've heard that I like is compositional coding. Marr and Nishihara here show how that would work for the human form that you could represent on the coarsest level of human form in terms of three-dimensional cylinders corresponding in length, width, and orientation and spatial arrangement to the torso, the head, the two arms, and the two legs. The reason that this idea has always been so theoretically appealing is, well, there are many reasons. One is that this represents a potentially terrific way of compressing information about something like the human form, originally represented in this megapixel image that's coming from the retina. That's a lot of signals. And it's way too many signals to store in memory. If you want to remember how somebody looked, it's way too many signals to send over axons of reasonable bandwidth. So I would say, even apart from invariance or stability, the bigger problem that the visual system faces is the need to compress things so that they can be stored in memory and so that they can be sent to other parts of the brain that need them. It's a very explicit sort of code, a code that would be easy to read. Information about the human form at the retinal level is extremely implicit. It would take lots of operations to pull out the information that there's a human form there. But this sort of code where you have individual signals for the shape of object parts, where they are relative to the rest of the object, how they're connected to the rest of the object. That's something that's very easy to read and makes it possible for other parts of the brain that need visual information to quickly extract the signal without having to go through all the nonlinear complications that the visual system does in order to make an explicit code. It achieves a lot in terms of invariance, and Anitha adverted to this and actually discussed some of the niceties of it. If you've got the position of all your signals specified in relation to a spatial reference frame that is centered on the object instead of centered on the retina as visual information initially is, then you've got automatic position invariance across the retina, and given the variation that Anitha gave evidence for, you've also got size invariance, because if your reference frame changes size with the object, then the positions of things in that reference frame remain the same. And then the other thing that's very appealing about this is the productivity of the code. So people have always worried about the fact that with a finite number of neurons, you've got to represent the virtual infinity of all the structures that you could ever encounter in life, or that I could make out of Play-Doh if I had a million years to do it. And the reason for this productivity is, in a sense, the same reason behind the productivity of just 26 letters of the Roman alphabet in different combinations can represent 5 million words in English and even more words in other languages. It's the combinatorial explosion of arrangements of those parts. This is my conclusion slide, and my main takeaway from this talk, or the takeaway I would like you to have, is the idea that the ventral pathway represents solid-shaped fragments for the purpose of compositional coding, and it represents those solid-shaped fragments in terms of parameterization of surface space, in terms of how surfaces are facing in three-dimensional space, what their surface normal orientation is, what their principal curvatures are and the orientations of those curvatures, and the position relative to the rest of the object of the surface fragments that they represent. And also in terms of medial axis shape. In a way, this is a false distinction, because medial axis is an abstraction from surface shape. Luckily, we had Jacob's talk yesterday, so you already know a lot about what is meant by medial axis. This is medial axis in 3 dimensions, which Jacob began to get into, and the parameters we can see tuning for are tangent orientation, curvature and the orientation of curvature, connectivity or topology, how different elongated elements are connected together, and again, object relative position. So in the rest of the talk, I will spend a good deal of time on new evidence from V4 for this, and then if I've got time, I'll go to older evidence in anterior of TE for this. We were surprised when we went into V4 and found that representation of solid shape rather than planar shape actually dominates as early as V4, which is just 1 synapse away from primary visual cortex. So area V4 is the first stage of the whole ventral pathway that's specific to the ventral pathway. V1 and V2 are omnibus areas that are sending information all over, all to all of the visual areas in the brain, and that includes visual areas in the dorsal pathway. V4 is the first stage that's really specific to the ventral pathway. It gets direct inputs from V1, so it's as little as 1 synapse away from V1. It gets massive inputs from V2. before that the receptive fields cover several degrees of visual angle near the fovea, and V4 is the primary source of input to the rest of the ventral pathway. So this is a real information bottleneck for everything. So this diagram, which you won't be able to see well, even if you squint, but this is a representation of the kind of experiment that we do in V4 and in further stages of the ventral pathway. It's an experiment in which the stimuli evolve in a direction that promotes getting more information about the tuning, the responses of a neuron in shape space. So this is an experiment on a single neuron at V1. We're using a microelectrode to record spikes or action potentials, and we measure the rate of spiking. This is in a monkey performing a fixation task to keep the eyes still. We start with initially random 3D stimuli, which again, I'm relying on Jacob here. Jacob showed you a generative model for medial axis shape, and our method for constructing the stimuli that I'll show you here is very much like that, defining the medial axis first in a probabilistic way, defining the surfaces around the medial axis in a probabilistic way. And then the way the stimuli evolve across multiple generations, it's controlled by a genetic algorithm where the fitness measure is just the neural response. So higher fitness stimuli, higher response stimuli are more likely to be represented by descendants in subsequent generations. The descendants are partially morphed versions of those ancestors, which represents local jumps in shape space. So in early generations, we're finding the general region of the tuning manifold for the cell, and in subsequent generations, we're sort of trying to pepper that region as closely as possible so that we can constrain a model of what information the cell actually represents. So again, that's not really visible. Here's the first part of the experiment. The whole experiment is divided into two lineages, lineage 1 on the left, lineage 2 on the right, and the reason for that is that we can then compare the lineages, we can cross-validate models, and we can prove to ourselves that the evolution doesn't just find a local minimum in response, or local maximum in response to space. It finds a global maximum that you can find no matter what kind of starting point you come from. And global maximum within the domain that we're able to create with our algorithm for creating shape. So let's zoom in even further, and I hope now you'll be able to appreciate the actual apparent three-dimensional nature of these shapes. We're using purely shading cues in our main experiment to convey three-dimensionality. What's shown up here is the stimuli in the first generation of lineage 1. These were all randomly created. You can see the variety of solid shape that we achieved, and they've all been ordered from top left to bottom right according to the response of the cell. And let me just check. We've got the cell here, the scale here. So bright red borders mean that the response was around 25 spikes per second, and black means 0 response.
This is a cell in V4. Yep. This is just one cell in V4.
Exactly right.
Picked at random.
You could say at random. We tried not to bias our selection at all, but we target V4 by various methods, and we find cells. We try to study every one. So here's generation 2. And generation 2, half of it consists of partially morphed descendants of mostly higher response stimuli up here. It's a probabilistic distribution that controls how we select ancestor stimuli. This high response stimulus here created these two partially morphed descendants right here. But then the other half of this generation and all the generations to follow involve taking mostly higher response stimuli, including this one, and then presenting them. Is this really in focus as much as it can be?
I can turn the light off.
No, it's okay. It probably is. So what we did was to take that high response stimulus, which we had only shown this way, show it again that way, but also show it in a different way. Here, the surface is virtually matte, so the only hues are shading. Here, the surface is virtually polished, so it has specular hues as well. And then we also show two-dimensional planar versions that are silhouettes that have exactly the same boundary contour but lack all the internal detail, and we show a bright silhouette and a dark silhouette. Lineage 1, generation 2, lineage 2, generation 2, and then what we've done here is for generations 3, 4, 5, 6, and 7, show the highest response stimuli and show an example of one of the stimuli that was tested in the 4 different conditions for both lineage 1 and lineage 2. And zooming in again, hopefully you can see the high response stimuli in lineage 1 across generations 3 through 7, and 3 through 7, and then from each generation 1 of the multiple stimuli that was chosen for presentation in these different conditions. And you can see in general highest responses for the matte condition, lower responses for specular, and mostly even lower responses for the two-dimensional versions. Two-dimensional stimuli, and I think Jacob may have even mentioned this, but it's something that's been studied by Peter Say. Two-dimensional stimuli like this, the boundary contour actually contains a lot of information about three-dimensional shape. So I'm not trying to say that two-dimensional shape is irrelevant or even unrelated to what we're looking at here. So it's surprising that we would see what seems to be selectivity for three-dimensional as opposed to two-dimensional shape in V4, considering that it's, again, only as little as 1 synapse away from V1. So we do a number of other experiments post hoc on these neurons to try to prove, this is when we have enough time to record and do these experiments, but to try to prove that this really is selectivity for 3D shape and not something about the lower level 2D nature of the images that we're showing. And one of the things we do, this is a natural question to ask, is there something about contrast relationships between stimulus and background in these stimuli that's evoking more response than the contrast we happen to be showing for the two-dimensional thing. So a way to look at that is just to do a contrast series for the three-dimensional shape and a contrast series for the two-dimensional shapes. And what these 4 examples illustrate, and it's a general result across all the cells we did this test, is that no matter how bright or how dark we made the two-dimensional stimuli, responses remain substantially lower than responses to the three-dimensional stimuli, even though in most cases those three-dimensional stimuli have a lot less contrast energy than the brightest two-dimensional stimulus. So far I've talked about conveying three-dimensional shape with specular cues and shading cues. Another kind of entirely different cue that we're actually very good at using to infer three-dimensional shape from an image is refraction patterns that you can see in like a glass sculpture, the way it takes things from the surround and distorts them. And then reflection patterns that you would see on a sculpture that has a chrome finish. You're able to appreciate the three-dimensional shape of that sculpture very keenly based on the way it distorts its reflection of the surrounding scenery. So we decided to try that on high-response stimuli for some of these neurons that showed selectivity for one kind of 3D shape and not for another kind of 3D shape. I don't know how well you can see it, but these are reflective and refractive versions of these shapes. And in general, the responses to the high response three-dimensional shape remained high, and the responses to low response three-dimensional shapes remained low. And of course, the actual image patterns that are involved in conveying three-dimensional shape here in the interior of the object completely different from what occurs in the shading and specular stimuli. So whatever this is robust across radical image changes. So highly likely to be related to three-dimensional shape, which is the one thing that's actually consistent across all these renderings.
I can't seem to see the difference between, besides the level of activations, but the actual stimuli, like the shape, they both seem specular. One looks like a three-dimensional Y, other one looks like a U. I can't seem to tell what's driving the difference in activations for the entire block on the left and not the one on the right.
So on the left, this is a shape that in the original evolution experiment, it evoked really high responses. On the right is a different three-dimensional shape that evokes low responses. So what we're showing is that these responses remain high, these responses remain low. Mainly this serves to rule out the possibility that refraction or reflection itself is driving responses. So then the gold standard for proving that you've got something that's depth related and not related to the 2D image is random dot stereograms.
So I'm just curious, in your previous original 3D experiments, the surface gradients are consistent with light coming from a certain direction, for example. So is this preference invariant to that sort of lighting condition? Have you tried different?
If I get to it, I'll show you an example of that very thing. It's not something we did a lot on in this experiment. So what we did with random dot stereograms, you've probably all seen these. It's like magic eye displays. You've got different displays to the two eyes. You're using the sensitivity to binocular disparity between the two eyes that underlies stereopsis, our appreciation of three-dimensional movies made by Marvel. We used that in this case to, in one condition, show a flat stimulus that had the same two-dimensional boundary contour as a high-response three-dimensional stimulus. In the other case, we used the disparities to actually create the appearance of a solid shape. In both cases, they're set against the background as well behind disparity. And in all of these experiments, what this graph shows is that the responses to the volumetric solid 3D shape stimuli remained higher than the responses to the 2D shape stimuli. 3D is yellow, blue is the planar stimuli. And I'm not sure why responses for these stimuli went down when, so the three conditions here are behind the fixation point, centered at the fixation point, in front of the fixation point, and I'm not sure why in front of involved lots of information, and if anybody has an idea, I'm willing to entertain it. Check it.
I don't have an idea, I have a question.
So I can't quite see it in the 3D cases there. The objects themselves are surfaced, textured with random dots, not just random.
Yeah, which is a little hard to do because, it can look a little bit flecky.
So you're varying disparity at sub dot size levels.
Right, yeah, so if you've got a shape that's going out like this, the disparity of points here are different from the disparity of points here. It's actually pretty compelling. So this is really the case where it's hard to argue that that the tuning of these cells doesn't represent something to do with volumetric solid shape, because there's really no image level explanation for this. So then I'll show you some analyses exemplified with this neuron that allowed us to figure out in a coarse way what aspect of solid shape was driving these neurons. If I get to stuff later, I can talk about more fine-grained analyses that are a lot closer to the kind of stuff that Peter talked about earlier this morning. But in this case, what we've done is a parameterization that's very close to our generative model in terms of the medial axes. We measure their three-dimensional position relative to the center of mass. We measure their orientation, and we measure the radius, which was another thing that we specified in our generation model, and then we used cubic equations cubic splines to smooth out the transitions between the different specified radii. And we get these sort of numbers for the shafts, the terminations, and the junctions between shafts. This is the highest response stimulus from lineage 1. This is the highest response stimulus from lineage 2. Just to adumbrate the result, you can see, and maybe you noticed this before, that the common geometric motif for the high response stimuli was this sort of dumbbell-shaped, bulbous-ended projection in a particular direction. I won't go into detail about this unless people want to, but whereas in other analyses we actually try to fit and verify a model, here we just wanted to do a spike-weighted average of the geometric measures we had to get, again, a coarse idea of what was driving these cells and whether there was some particular thing driving these cells. So each of these stimuli, based on the parameterization that I just diagrammed, corresponds to a set of points in a multidimensional space where the dimensions are the orientations, the positions, the connectivity, etc., of the shafts and the terminations and the junctions. And so we find those points in the space, we smooth them with a Gaussian, and we weight them by the response of the neuron to that stimulus. So this stimulus has a high weight, so it's going to contribute a lot of energy in regions that correspond to, say, the termination here, the termination here, and the termination here. This also gets a pretty high rate, and it's going to contribute energy to parts of the matrix that correspond to this termination, this termination, this termination, but also this termination. So these two are adding up. These lower response stimuli will contribute very little. We sum all of those together. And then we do the same thing for lineage 2, and then we take these two matrices and we multiply them together. And that's a way of emphasizing the geometric motifs that appeared in high response stimuli in both lineages and de-emphasizing or taking out things that rule along for free that happened in one lineage to be correlated with the high response shape. And that gives us a final result that we can break out into separate diagrams to highlight the nature of what seems to drive the cell. Looking first at terminations. So this is a spherical plot of termination angular position, that is the position, the angular position of the high response driving terminations relative to the object center. So object center would be at the center of the sphere, and this direction corresponds to the top of the object, the bottom of the object, the back of the object, the front of the object, the left and right. And the reason this is all turned around is just so that we can get a good look at the peak. So yellow represents the peak on the scale, and blue represents the bottom. And what this shows is that the terminations that drove high responses across many stimuli were positioned to the bottom and to the left relative to object center, which you can see there. And then this is a polar plot that represents termination direction. So now instead of position relative to center of mass, different points along this sphere represent the direction in which a termination was terminating. And again, top is up here. Now front is here, left is here, right is here. And this peak corresponds to the bottom left of the entire sphere of possible termination directions, and you can see that that's the direction in which these high response stimuli have terminations pointing. And then the last really relevant thing that comes out of this is tuning for termination radius. So low responses when radius was small, high responses when radius was large, hence or that reflects the bulbous nature of these terminations. And then the other parts of these stimuli that define high responses were these shafts leading into these terminations. Of course, these are highly correlated in our stimulus set, and in the natural world, there are naturally going to be high correlations between parts of shapes that are necessarily connected. So for shafts, the position is again behind and to the left, and shaft orientation, which doesn't have a directionality, so it's only a hemispherical surface, is up here. So that really corresponds to the same thing that we saw for this termination. It's just that this is sort of the other end of a line going through the center of the sphere. And then the last interesting thing about the shaft specification was that now responses were highest for thin shafts and went down as shafts got wider, so which represents the sensitivity of the neuron to this radius difference between this part of the shaft and the ending. And so that's a cheap, low-dimensional way of entailing some information about the surface curvature here and sort of the growing radius towards the end here. The reason for using this cheap, low-dimensional analysis is that it makes our analysis more powerful, and the more dimensions you have, the more data you need to constrain a model.
So, sorry if I missed this, but what's your measure of eccentricity?
It's distance. So
Oh,
the
it's a
overall.
global eccentricity. It's not a local curvature measurement.
Yeah. Sorry. Eccentricity, distance away from the center of mass.
Okay.
So these are all the plots I've already shown you, and even though we weren't trying to make a model, we're claiming that this is something about how V4 is really working. We did look at how good the predictions based on this spike-weighted average could be relative to the observed responses, and they're pretty good. And for this neuron, because it cared about terminals and shafts, the predictions were best with a linear combination of the two. And then looking across an entire population, here is the correlation coefficients observed versus predicted responses based on shafts, based on junctions, based on terminations. These are all actually pretty absurdly high correlation coefficients, but of course this is not cross-validated here. This is fit to the entire set. All of these are getting high correlation coefficients, which of course represents the fact that they're capturing much of the same variance because they're highly correlated within the shapes. Completely different kind of experiment done in the laboratory of our collaborator Christina Nielsen, experiments based on photon imaging in anesthetized monkeys. And here are 6 different neurons and their change in fluorescence, calcium fluorescence, which is a proxy for neural activity across time following stimulus presentation. In this experiment, we used 3-dimensional shaded stimuli as well as flat, bright, or dark 2-dimensional stimuli. And this neuron is demonstrating that there's a substantial increase in neural activity as reflected by calcium fluorescence for the 3-dimensional stimulus, not for the 2-dimensional stimuli. And then there's some neurons that have equal or stronger responses to 2-dimensional stimuli. So here's the actual image that came from. Each of these neurons is identified by a couple of tests as a neuron, and then the color code represents the bias towards solid shape, where dark purple, which really doesn't occur here, would represent responses to purely 2D shapes, no responses to 3D shapes. The blue that you see through most of here is a weak preference for flat shapes, and yellow and orange is preference for 3-dimensional shapes. One thing that you can see, and there's an analysis behind this, but I won't go into the detail, is that the solid shaped neurons and the flat planar shaped neurons cluster. Clustering across the cortical surface is a major signature of important tuning dimensions throughout primate cortex. It really is a cluster because if you define the boundary mathematically and you look at distances of neurons relative to the boundary, and you look at the solid preference score as you go basically from here to here, you really see a step function. It's not simply a gradient between one or the other. And so that's one example. There's another example. There's another example. There's another example. And the general finding was that if you look at a cumulative histogram for this bias towards solid shape, you see not a whole lot of bias towards planar shapes, plenty of bias towards solid shapes. I've only got a few minutes, 2 minutes. Let me just jump through this. TE, same kind of experiment. Here, we really did modeling. We broke up the surface into relatively consistent surface fragments using something similar to what Peter was talking about, and then fit a model based on those fragments to describe what was the common feature in all these high response stimuli for this TE neuron. One thing about TE that's different from earlier stages is that instead of representing single parts, neurons represent configurations of multiple parts. This is representing these ridges facing towards the viewer and these fairly flat dorsal surfaces. this is what Anita asked about, what happens if you change the lighting angle, which is another way to radically change the two-dimensional image and rule out simple explanations like, oh, it's just like something dark below something bright. So these are the responses of this neuron as you go, as you change the lighting direction in the vertical and change the lighting direction in the horizontal. You can see how different the brightness pattern here is, but the response is very much the same. That represents a sampling of the different kinds of sensitivities to surface shape we got. I just want to throw this in really quickly because Jacob talked about medial axis. We've also done in TE experiments like this that are more focused on medial axis shape. Here are the last couple of generations in lineage 1 for TE neuron, the last couple of generations in lineage 2. Here we are modeling medial axis sensitivity with a template procedure which perhaps could have benefited from Peter's idea of calculating the gravitational attraction between two templates. But the important point here is that even though these neurons are representing more complex configurations of multiple shaped parts, it's still the case that they're not representing an entire shape. They're representing parts of shapes, and just as in before, you can throw a lot of other stuff into the rest of the shape, and the cell doesn't care. There's some cells, like this one, that seem to mainly care about medial axis shape, and as a result, if you keep the same medial axis, but then change the variation in cross-section of the surface shape that surrounds it, you get pretty high responses here, around 60 spikes per second, regardless. But most neurons in TE are actually sensitive to both medial axis shape and surface shape, and it's only when both are right that you get high responses. This is a bunch of different neurons with the same kind of analysis. This is my conclusion slide again, and these are people who did work. The guy who spearheaded both the neurophysiology in V4 and the two-photon in V4 is Ramanujan Srinath. Alexandria produced the blender-based images of refractive and reflective shapes. Alice is doing some deep network-related analysis using ROM's data. Yukako did the surface experiment in TE, and Alden Hung did the medial axis experiment in TE, and Christina Nielsen is our collaborator who is an expert in primate. Thanks.
Questions? Peter. So I'm curious how you mentioned breaking up the surface into fragments, and that's something we've been wrestling with. We don't have a good solution. How are you doing this?
So I guess what you would call a 3D algorithm. We tried to grow the largest regions we could, but we had constraints on how much the orientation could change, the surface orientation, and how much the curvature could change. So just as I think you showed, chopping things up based on inflection points. This is chopping up based on inflections as well as, sharp, high derivative curvature. And I don't have the figure, but I'd be glad to correspond. I think it worked pretty well. It gives you exactly sort of the things you would name as parts of the surface.
Sorry.
I was wondering, how would you rank the contributions of different 3D elements such as shading, illumination, refraction to V4 coding?
Yeah, great question, and something we've addressed in other experiments. We haven't addressed a whole lot in V4. It depends on the neuron. So in TE, it's actually here. We did just the silhouette stimulus with no
3D.
with no 3D at all. We did just a texture. We had spots, so you had texture gradients. We did shading alone, and then we did the same things, but we added stereo. And whether there was only stereo or stereo with texture, stereo with shading, great responses. Even without stereo, shading does pretty well. We, on the other hand, identified a channel in TE, which to our surprise was actually much more sensitive to really large-scale scene-sized shape. And there, probably because stereopsis is only useful out to about 10 feet, and after that, shape does. There's very little difference between the different parts of the shape. It's really texture flows that are the big Q, and disparity makes very little difference. Shading has some effect. I don't have a good answer for D4.
If you plot the union or maybe the sum of all these tuning curves in the parametrized space of shapes that you have, are there any irregularities, or do they look completely kind of spread out?
Oh yeah, that's something we've been very interested in, and it would take a long time to completely answer, but there are lots of anisotropies in those plots. One of the most striking ones, this was based on the analysis of two-dimensional shapes before, is that there's a huge bias towards curvature extrema, which is another thing that has come up repeatedly during this conference. And then if you fit a network model to try to discriminate between the boundaries of lots of natural shapes, it doesn't look like that at all. In fact, all the energy is concentrated towards zero curvature, fairly flat parts of the boundary. But then if you put a sparsity constraint on the network as well, everything migrates up to this. So there's actually an 8-fold change in the amount of neural activity used to represent a boundary shape if you do it in terms of the curvature extrema instead of flat portions. So that's part of the compression theme. I mean, it's an 8-fold compression of that kind of information.
Last question.
So is there a time course difference in terms of selectivity for 2D versus 3D shapes in your V4 data? Does 3D selectivity come up later?
I don't have that answer before, but we looked at it carefully in PE, and to my chagrin, there was none.
Oh, yeah?